it's very nice to have our second talk of the school. And well, first of all, I want to thank Pablo and everyone at ICTP for putting the school together. It has been fantastic. The work that they are doing was just amazing. I would share applause for them. Please, let's join us too, because you have no idea the amount of work involved on this. So thank you. And well, for the second lecture today, we have the honor and pleasure to have Olga Dudko speaking to us. She's a professor at UC San Diego. Olga is from Ukraine. So we are so supportive. We have no idea what you've been through, and you were fantastically brave to stand to the right things that have to do on these days. So it's very nice. So Olga, it's not the first time she's been in Brazil. She's been here before, and well, so she got her undergrad and graduate studies back in Ukraine at Kharkiv, right? And then she was in Israel, and then went to United States, where she was at NIH, working in Watzlaw and Hummer, and then from there she went to CSD, where she's been faculty since 2007. And she's a professor there, and she's a fellow of American Physical Society, and she has had a long trajectory doing on condensed matter physics, but these days she's doing biological physics. And Olga, please, your stand is yours. Thank you so much.
Good morning. Thanks very much for the kind introduction. Thank you for inviting me. I look forward to talking to all of you during the lecture and also outside of the lecture. So the organizers kindly invited me to talk to you on the physics of life over the course of a few days. And my plan is to tell you about some what I think are truly fascinating topics that are actually have little in common in biological sense. But they all have some common threads. And one of these common threads is the notion of time, how living systems manage time. As I'm sure you know, often organisms, cells and organisms are not satisfied with intrinsic rates of underlying processes, for example, chemical reactions. They just go too slow for a particular biological function. And so living systems find ways to alter those rates, for example, to speed them up, sometimes by many, many orders of magnitude. Of course, you're aware of the enzymatic catalysis. By employing enzymes, cells and organisms can make reaction rates go much, much faster, right, or sometimes slower. But there are other examples. For example, when diffusion is too slow to serve as a mechanism of moving things from here to there inside a cell, then cells can recruit molecular motors and speed up the transport rates by orders of magnitude. But even if the motor-mediated transport is too slow, you can also use biological electricity to speed up the rate at which information is propagated by many more orders of magnitude. What I would like to us to think today about is the case where living systems manage to accomplish things on time, even though at first it may seem impossible, by a clever use of general physical principles. Specifically, I would like us to talk, to think about how remote gene segments find each other in the crowded environment of the cell nucleus. So we humans are eukaryotes, which means that our cells have, each of our cells has nucleus, almost every cell has nucleus, and so the genome is stored inside that nucleus. A typical size of the nucleus is about 10 microns in diameter, and so the entire genome is stored there. So if you take DNA, your entire genome, and extract it from a single nucleus of 10 microns in diameter, unravel it, and fully stretch it, so you end up with double helix, right? How long will that polymer be? Does anyone remember, or more interestingly, would anyone like to guess? So we have DNA stored inside 10 microns, container, and we take DNA out, we, get rid of all the packaging material, proteins, and so on, and fully stretch it, so we end up with the entire genome, double helix. How long is that? Order of magnitude? Couple of meters. Yes, exactly. Yeah, isn't it stunning? So we have 2-meter long polymer that is stored inside a container of only 10 microns in diameter. So it really is this stunning degree of compaction. And the question is, how does it all fit there, right? Surprisingly, to me at least, we actually know very little about how DNA is packaged inside the cell nucleus. We, of course, know the structure of the double helix. We know that at the most basic level, DNA does not, by the way, exist in nucleus in its bare form. At the most basic level, DNA is wrapped around proteins called histone octamers, to form a configuration that is often referred to as beads on a string configuration. So those beads are so-called nucleosomes. And each nucleosome is essentially a macromolecular complex which consists of a protein histone octamer, right, with DNA wrapped around it. So there are about 2 turns of DNA wrapped around each histone octamer, and that's a nucleosome. So this bead on a string configuration is what is usually referred to as 11 nanometer thick fiber, right? It's a thin chromatin fiber. But how is this fiber packaged further to fit it in the nucleus is still largely a mystery. It is especially a mystery during the phase of the stage of the cell cycle known as interphase. So in general, the density of chromatin fiber depends on the density of DNA packaging. It depends on the stage of the cell cycle. And so during the interphase, the chromatin is in a relatively decondensed phase. And the fact that it's decondensed makes it particularly challenging to try to visualize how it is packaged. So essentially, all the standard biophysical methods that are so remarkably powerful in visualizing what's inside a cell are essentially blind in the sense of trying to get the idea of the organization of chromatin during the interphase. However, it is during the interphase that it is particularly important to understand how DNA is packaged in the nucleus, because interphase is a stage of the cell cycle when the genes are actively transcribed. This is a stage of the cell cycle where the productive genomic interactions occur, right? And so it is important to know how DNA is packaged. Now, to add to this already complicated story, chromosomes are not static occupants of the nucleus. They constantly wiggle around. And so whatever the folding pattern of a chromatin is, it changes with time. Now, why is it important to understand how the 3-dimensional structure, I should say 4-dimensional structure, 3-dimensional spatial structure plus the temporal dimension of DNA inside the nucleus? First of all, obviously, it is an interesting physics problem, right? How 2-meter-long polymer is packaged inside a space of 10 microns in characteristic size, but it also is a crucial biological question. And the reason, it's very easy to see why. So imagine 2 DNA segments that are marked by bright spots here that are far apart along the contour lengths of DNA. But in a three-dimensional space of the folded chromosome, they end up neighbors. And so imagine that one of these genomic segments is an enhancer and the other one is its target promoter. So their interaction in 3D space may trigger gene expression, which in turn may have crucial consequences for the fate of the cell and of the entire organism. Or these two segments may be two segments in the locus, in the region of DNA that encodes antibodies, right? And so as we will talk about in a few moments, antibodies are coded by genes which themselves are made of gene segments, and so the process involves remote genomic segments, remote in the sense they're far apart along the DNA, finding each other in a 3D space of the nucleus and interacting. And so the interaction will create a gene that will give rise to an antibody, which may also have crucial consequences for the cell of the organism. Now, if you look at the classical textbooks, you actually may get a completely different impression from what I just told you, including what I think is a really excellent textbook from which I personally learned a lot, Molecular Biology of the Cell. It tells you that the way the DNA is packaged inside the nucleus is that the double helix undergoes several levels of compaction, a hierarchy of compaction, where we start with this bead on a string configuration that I just described, where DNA is wrapped around histone octamers to form nucleosomes. And then the textbooks say this 11 nanometer thick chromatin fiber is folded further into what is known as the 30 nanometer thick fiber. And then the 30 nanometer thick fiber forms loops that allow the DNA to be packaged in this very small volume of the nucleus. The interesting thing is that despite what all the textbooks tell you, no one has ever seen the 30 nanometer thick fiber in live cell. People have seen it in vitro experiments where they extracted chromatin from the nucleus and used, for example, atomic force microscope to stretch it. And then they could see what they call the 30 nanometer thick fibers. Then if they pull further, the 11 nanometer thick fiber. But no one has provided evidence of that the 30 nanometer thick fiber exists in vivo. And here's just one example of relatively recent studies where people used a combination of two techniques, chemical labeling and electron microscopy, to try to peek inside the nucleus and determine the organization of chromatin, and in particular to see any hint of the existence of this structure from the textbook. And no, they haven't found it. What they found is that chromatin is a disordered, the vast majority of chromatin in the nucleus is a disordered polymer with varying diameter in the range between 5 and 24 nanometers, and no evidence of the existence of the 30 nanometer thick fiber has been found in living cells. So to make things a little bit simpler and more manageable, here's our puzzle. The puzzle is that we have a very long polymer that is packaged in an unknown fashion inside the nucleus, and we are interested in how the segments of DNA that are separated by vast genomic distances along the DNA find each other inside the nucleus, and how they find each other on a very short time scale, the time scale that is meaningful from the point of view of biological function. There are several reasons why this question is non-trivial, not just because the two segments are far apart along the DNA, along the contour length of the DNA, but also because they move not through a theoretical physicist, like perfect homogeneous aqueous environment, they move through a very crowded environment of the cell nucleus, and they encounter all these obstacles from chromatin fibers that we don't even know their structure. So what are the possible answers to this question? So can we think about hypothetical solutions about how remote DNA segments manage to find each other quickly despite all the complexity that is happening inside the nucleus? I would like to hear your ideas, and I'm not talking about anything detailed, just in wide brush strokes, some extreme scenario. What can you imagine?
Some sort of long-range interaction?
So we are asking about the long-range interactions between the two segments. Sorry, so are you suggesting that the elements, as a possibility, right, as a possibility, that the two elements sense each other? That certainly something that one can imagine, but let's also think about the fact that there's so much in between, right? All the chromatins and proteins, and probably even if such strong interactions existed, they would be screened, right? Yes? Sorry, chemical interactions. Well, I think it's a similar idea, right? And most definitely there has to be some chemical interactions, but again, the interaction starts to play a role when the two segments are already near each other, right? But when they are far apart, right? So how do they get into the same proximity within the nucleus? There was another hand, yes. Yes, please. Yeah, I'm asking a question, you're responding with a question, yes.
How unique are these segments?
How what?
How unique are these segments? Are there many of the red segments around?
Yeah, it's a good question, which goes back to the idea of specific interactions between them. They are quite unique. I mean, it depends, I guess, on the context, on the biological context. There are contexts where you can choose among, let's say, a few hundreds, right? There are contexts where it is a particular enhancer and particular promoter, right? So these are truly unique. So there is a range. Yes? Why do they have to be quickly? Why do they have to?
What does it mean to be so quick?
Sorry, what?
What does it mean that it is?
Oh, what it means quickly means on a time scale that, so let's say you are stressed and you need to produce some protein to help you handle the stress, right? So if it takes you, hours or days to produce that protein, in other words, you are giving these two segments hours or days to find each other, that's not very helpful, right? So quickly means on a time scale that, again, depend on the context, but, minutes, under an hour, right?
So here, up here.
Yeah.
So I'm not very familiar with this topic.
That's
yeah,
especially
so
easy, yeah.
are we assuming that some of these long-range interactions happen regularly and not at random? Because my first thought, and maybe it's a little bit simplistic, is that the whole DNA, the double helix, would be just moving at random and eventually creating these long-range interactions. But this would just lead to random interactions and not regular interactions that are happening all the time, if that makes sense.
And so if it's random, then what?
Well, if it's moving at random, then we could, at different times, we would have different interactions between distant DNA segments. And yeah, but like, I guess that the way you're framing this question is, how do these different segments find each other? So what I don't understand is, do they find each other as?
By finding each other, I mean they end up in the same place in a relatively short amount of time so that they can actually use their chemical, interactions to do something, to start producing a protein or to recombine to form a gene out of segments, right? But I like that you introduced, maybe I would love to hear more suggestions, but maybe we should move on. You have suggestions?
Oh, what
Yeah,
about?
you're not assuming?
Yeah, well, just
Please,
borrowing
please. Yes.
the information from protein folding. It's just
Exactly,
that
yes.
you have the energy landscape that, given the environmental conditions, it favors that particular state in a long range,
Yes.
right? So it's not going to be a totally random
Yes.
type of search.
Yes, indeed, not totally random, but I like how you introduced the idea of randomness. And so I think from what I heard, we actually have sort of two maybe extremes, right? One extreme is I think there's some resonance here, so I shouldn't get too close. One idea is that there are some tightly regulated biological processes that deliver the segments to each other. We can even imagine some kind of molecular motors that move these two segments. Perhaps there is some gradient that follows those chemical interactions that have been mentioned. And on the other extreme, we can imagine that things happen completely randomly, right? Through just some general physical processes. For example, we just let the two segments diffuse, and somehow, miraculously, they will find each other on a sufficiently short time. And of course, there are many possibilities in between or combining these two. So my group has been fortunate to study this question in a fascinating context, the context of adaptive immune system. So let me say first a few words about what my biology colleagues have been measuring, and then we will make sure to discuss how is this related to the question that we posed, how remote genomic segments find each other in the nucleus. When a cell or an organism is infected by a virus, the infection triggers immune response. And as you, of course, know, some of the key players of your adaptive immune system are B cells. B cells produce antibodies. Antibodies bind and inactivate antigens. And quite remarkably, and this is the power of our adaptive immune system, there is essentially unlimited number of different types of antibodies that our immune system can produce. But wait, antibodies are proteins. Proteins are encoded by genes. And our genome definitely does not have a virtually unlimited number of genes, right? So how is it possible that our immune system can produce many more different types of antibodies, proteins, than there are genes in our genome? And the answer to this question is the mechanism by which the genes that lead to the pool of antibodies, these genes themselves are assembled. They are assembled from gene segments. Okay, so the genes that encode antibodies are assembled from gene segments, and the process through which this assembly of genes out of gene segments occurs is called VDJ recombination. And just a very schematic view of the idea, what does this VDJ recombination mean? So here is a region of DNA called immunoglobulin heavy chain locus, which is simply a stretch of DNA of about 3 million base pairs long. And this is the region of DNA that codes for antibodies. The locus consists of multiple copies of so-called V regions. V stands for variable, and by multiple I mean a couple of hundred copies. It contains multiple copies of D regions and multiple copies of J regions. D stands for diversity, and J stands for joining. So VDJ recombination occurs in two steps. In the first step, one of the D segments recombines with one of the J segments to form a DJ joint. And in the second step of VDJ recombination, this DJ joint recombines with one of the V segments to form VDJ gene. Okay, so this is a gene that will give rise to a particular antibody, and because of the combinatorial nature of this process, right, any of these V's can recombine with DJ, and also any of the D's can recombine with any of the J's. Because of the combinatorial nature of this process, the result is a diverse pool of antibodies. And this, there are actually mechanisms that amplify the diversity of this pool even further, but at least on the most basic level, the reason that we can make so many more different types of antibodies than there are genes in our genome is the combinatorial nature of the process by which these genes are assembled, the process of VDJ recombination. Now, you can probably recognize in this process these scenarios that we've discussed at the beginning. So in order for this to happen, two genomic segments that can be very far apart along the DNA, by far apart I mean up to 2 million base pairs, DJ and one of the V's, right? They have to find each other to form, to recombine, to form a gene. And there was a question, what does it mean to find each other quickly? What does it mean quickly, right? Well, quickly enough so that you can produce antibodies in time to fight the infection. So under an hour, presumably, would be something reasonable. So here is an example of a context where the question of how remote genes find each other is the crucial question. So the first breakthrough in addressing this question in the context of VDJ recombination was made some time ago by awesome biologists who later became our collaborators. What they did, they were able to visualize the motion of genomic segments inside live mammalian cells, B cells, from mice. So just to make it more clear what we are looking at the nuclei from B cells, and two green dots mark the location of the same segment, DJ segment. That's a DJ segment that is formed after the first step of VDJ recombination on two sister chromosomes. Okay, so Joe Lucas, a former grad student in the lab of my biology collaborator, Kees Murray, developed a genetically modified mouse which carried in its genome two fluorescent markers, green markers, that marked the location of DJ segments on two alleles, on two sister chromosomes. And this allowed them to visualize the motion of the labeled genomic regions in real time inside living cells. Now, of course, the real question that they and we were interested in is how each of these DJ segments, each of the green dot, interacts with the V segment, with any of the V segments inside the same chromosome. But answering that question directly, visualizing that, would require a second color, right? So at that stage, doing the single color experiment was already, it's an incredible breakthrough that took Joe, I guess 4 maybe years at least to make it work, because the standard techniques, labeling techniques that work for cells like yeast, for example, they do not work for B cells.
Can I ask you something?
Yes.
But the D and the J also have to find.
No. So they're on different chromosomes, right? And they do not interact. And by the
They are together, the D and the J.
sorry,
But the D and the J are already together, or they have to
V and DJ.
No, D
Oh,
and
DJ.
J.
Yes. So DJ.
They are always together.
So this experiment was done after the first step of VDJ recombination, and the first step, remember, is D and J recombine. So we are now interested in the second step, where recombination of D and J is interesting, but it's not that surprising because they are relatively nearby along the DNA.
But what do you mark then? Or is
So
it
what is
you mark?
marked is after D and J recombine, so you have a DJ joined, and this is what you mark.
Okay. So
Right?
you need them to be together to be able to observe them. It's not that you can see D and J separately where they are all.
You could, but that would be a different experiment, right?
Okay. But with this marker, you only see the D and J when they are together.
It's not about the marker, it's about when you mark it, when you tag it, right? And so
Okay.
you insert the tag after D and J have recombined.
Okay.
So you have a DJ segment.
Okay.
joint, right?
Thank you.
Now, what you're interested in is how this DJ joined interacts with V in the same chromosome, but you can't see V. You would need a second color, and I'll tell you about the second color maybe actually next time, right? So you asked about the two DJs on two chromosomes, they do not interact. And by the way, this is true in general. People notice that there's very little interactions between segments on different chromosomes. Most of the interactions occurs not just within the same chromosome, but within a particular chromosomal domain, right? But this is what was possible to do at that stage, is to mark two DJs on two sister chromosomes, right? Each of these DJs will have to find its V within its own chromosome, but we don't have a second color at this stage, which means that we have to rely on a theory, right, and simulations to make predictions. So it was a very timely question. So what we want to predict is how V might be moving relative to DJ based on what is observed. And what is observed is how DJ moves relative to DJ on two different chromosomes. So again, it was a remarkable breakthrough, but not quite at the level where we can directly visualize two genomic segments moving within the same chromosome, right? Right, so this is something we would have to figure out. This is an experiment, an example of an experimental output. So what was measured is three-dimensional trajectories of each of the fluorescent dot, in other words, each of the DJ segments. And it is important that these are 3D trajectories, because from the video it may not be obvious, right? These are 3D trajectories, so you have X, Y, Z as a function of time for each of the DJ segments. So what do you think? What kind of motion is this? I mean, clearly this is not something that would fit a scenario where molecular motors would move persistently in a particular direction, the two segments toward each other, right? So this looks more diffusive like diffusive light trajectories. But the question is what kind of diffusive motion this is. Some of the questions that one may try to pose, at least at this stage. So first of all, the question of the mechanism of DNA motion. And as physicists, I was told that most people here are physicists, so I don't need to say that when we say mechanism, we really are looking for something quantitative, right? I mean, an equation, a law, right, that governs the motion of DNA segments. And once we have an equation, we can, well, maybe at first we should try to reproduce what is observed, but then importantly, we should try to make experimentally testable predictions. So, mechanism of DNA motion. We want to know how do DNA segments find each other given that mechanism that we will have hopefully established. We want to know what parameters, what properties of the of chromatin, of nucleus, of something else, what properties control the time scale, and what is the time scale, right? How fast do they find each other? And also, it would be nice to have an equation that predicts the time scale based on the parameters of the system. So when we have diffusive-looking trajectories, there are a number of, I could call them diagnostic tools that are very useful to try to extract the mechanism of motion from the trajectories. And the first such tool is something that you, of course, know very well about, is the mean square displacement, MSD. Intuitively, mean square displacement is the measure of the amount of space that is explored by a random walker over time tau. And in this cartoon by George Gamow, the random walk is represented as a drunkard's walk. And so we can imagine the following experiment. We can put the drunkard at the origin, this street light, and let him perform a random walk over a given time, let's say tau, right? And by the end of that random walk, we can measure his end-to-end displacement, right, R. But in the true spirit of physics, we should do this experiment many, many times. So we return the drunkard to the origin and let him perform another realization of the random walk over the same time tau, and then measure the end-to-end displacement again, and do this many, many times. Alternatively, we can take a statistical ensemble of nominally identical drunkards, release them all, they're non-interacting, of course, release them from the origin, and let them perform their random walks over time tau, and then measure the end-to-end displacement for each, right? And hopefully that will give us some sense of the amount of space they have explored, even though their walk seems purposeless, and it is probably, but it does not mean they do not get anywhere, right? They do explore the space around the streetlight. And so what happens if we do this experiment and then take the average of R? What shall we get? The average. No. This is statistical ensemble, yeah. So we do this experiment for many identical drunkards, and by the end of their respective walk, we measure the average of the end-to-end displacement. The average R, not yet the mean square displacement. Of course, it should be 0, right? That's a random walk after all. So indeed, the average on average R is 0, and so this is why the mean square displacement is a good alternative, a more meaningful, so more informative alternative of the measure of the extent of excursions of the random walker, right, during time tau. So let's now define mean square displacement. We can define it, we can compute mean square displacement in more than one way. We can compute the ensemble averaged mean square displacement, and this is the experiment that I just mentioned with a statistical ensemble of non-interacting, nominally identical drunkards. So to calculate ensemble averaged mean square displacement, we first compute the squared displacement for each random walker during time tau, and because it's squared, we no longer have this problem of average being 0, right? And then, this is what the angular brackets do, we average over the ensemble. So it's a mean square displacement averaged over the ensemble. There's another approach to calculating mean square displacement. We can first calculate the squared displacement for a single random walker during time tau, and then average over a long period of time. And the procedure of averaging over time is right here. So we are integrating over time between 0 and t. Capital T is the observation time, divide by capital T, and take the limit in which capital T becomes very, very large, right? So this is the time average mean square displacement. And of course, we can calculate time and ensemble averaged mean square displacement, where we can do both type of, we can first calculate the square displacement for a single walker, and then we do both types of averaging over ensemble and over time. Now, I have a question to you. So ensemble averaged MSD and time averaged MSD, are they the same? Should they give you the same answer, or this is actually a physics question, or different answers, or it depends? What do you think? So we don't know yet, right, what will happen if we use these two, calculate these two types of MSD on the actual biological, the trajectories of genomic segments. But in principle, in general, ensemble averaged mean square displacement and time averaged mean square displacement, do they have to be the same? Can they be the same? Please. Yeah. Sorry.
No, I would hope they coincide if the system is encoded.
Yes. Yeah.
I
I'm assuming everyone heard, right? So the answer was that if the system is ergodic, then indeed we expect the time and ensemble averaged MSD to be the same. Excellent. And for normal diffusion, that would not be a surprise. This is what we would expect. But for the diffusive motion of the gene segments, we will have to check if it's normal diffusion or if it's some weird diffusion. And if it's a weird diffusion, then it actually is a very interesting question. is the process ergodic, right? And so we, right here, in front of us, we have a criterion. From the trajectories, we can always compute mean square displacement averaged over ensemble, separately averaged over time, and then compare the two. Okay, so for a broad range of systems, mean square displacement has a particular scaling with the time lag. You know, tau is the time you let your random walker to explore space. And the scaling is here. Mean square displacement scales with time as time, tau, to the power of alpha. Just from the form of the scaling, it is immediately clear that the best way to plot mean square displacement as a function of time is on a log-log plot, right? Because the scaling on a log-log plot will result in a straight line, a linear graph, and you can immediately extract the value of alpha from the slope. It's obvious, right? Now, alpha is the scaling exponent, and it is actually incredibly informative. So scaling exponent alpha tells us something about the type of the underlying diffusive motion. Alpha equal to 1 recovers the famous result from the classical diffusion theory that states that mean square displacement scales linearly with time, right? And this is what you expect from the classical, experiment of pollen grains diffusing in water that has, if I remember correctly, has led to the discovery of Brownian motion. Now, there is a possibility that alpha may be not equal to 1. And alpha not equal to 1 corresponds to anomalous diffusion. Specifically, alpha greater than 1 is called super diffusion, and alpha less than 1 is called sub-diffusion. Okay? So alpha tells us about the type of diffusion. Alpha equal to 1 is normal diffusion, and it's always useful to compare whatever results we have to that case of normal diffusion. It's a good reference point. And alpha not equal to 1 is anomalous diffusion. So we can see that for the case of alpha less than 1, this is the subdiffusive motion, obviously mean square displacement does not grow as fast with time as the normal diffusion does, right? Which means that if we discover, for example, that our genomic segments move subdiffusively, we expect them not to go as far as they perform their random walk as a normal diffusing random walk would do, right? Which seem to suggest that there are some disadvantages of being a subdiffusive random walker, right? Because if you're looking for your partner genomic segments that is, maybe, relatively far apart, then if you can't go as far during time tau, then perhaps your chances of finding that partner segments are not as high, right? However, let's look at these cases of superdiffusion, normal diffusion, subdiffusion at the level of the actual trajectories, the microscopic trajectories. So if you zoom in and examine the trajectories, you'll find that for a superdiffusive motion, the trajectories tend to be more smooth than for normal diffusion. And on the other extreme, for a subdiffusive random walker, the trajectories tend to be much more rugged. Well, depending on how small alpha is. The small alpha, the more rugged the trajectory is compared to a normal diffusion. So can we now quickly revisit my statement from a few moments ago? I suggested that just by looking at the way that MSD scales with time, a subdiffusive motion seems to be have disadvantages from the point of view of biology, from the point of view of searching for a partner segment, because well, you can't go as far, right, in a given amount of time as normal diffusion would be able to. But just by looking at the trajectory and noticing that its ruggedness compared to the normal diffusion, can you think of the pros of being a subdiffusive random walker from the point of view of finding your partner gene, genomic segments? Please. Yeah, just a second. Microphone is coming.
So, well, I can try to answer a question, but I also have another question. I mean, if we want distant DNA segments to find each other quickly, I assume the super diffusive behavior would be better because it just leads to bigger displacements in a shorter time.
That actually is another way of answering my question.
But
because by that logic, subdiffuser will not be as good.
Yes. But at the same time, I'm wondering, sure, the segments need to find each other, but for how long do they need to interact with each other? Or do they just need to find each other instantaneously and then they can
No,
drift
that is
apart?
indeed a relevant question. Once they have found each other, they need definitely to be colocalized in space sufficiently long to interact. Yeah, it's a good point. But for now, we are just interested in that first stage of them finding each other in this insanely complex environment of the nucleus. So my question was, can you think of any advantages of being a subdiffusive random walker, and therefore your MSD does not grow with time as fast, obviously, as normal diffusion, right? However, your trajectory is much more rugged. Do you think there's anything good associated with the ruggedness of the trajectory from the point of view of finding your partner gene segment?
Doesn't it all depend on where the partner is? Because if you have a more complicated trajectory, you are exposed to more genes.
It's not that it's more complicated, it's more rugged, right? So
You could explore more.
you search more thoroughly,
Yeah.
right? Exactly. Yes, this is an excellent point. So right, yes. So on the one extreme, if you're a super diffuser, right, so your trajectory is smooth, it means that your MSD grows very quickly with time, but the way you search, because of your smooth trajectory, you check here, and then you check here, and then you check here, right, but you're missing everything in between. However, as you just suggested, with the trajectory that is rugged like this, you don't explore too far, right? But you check, here and here and maybe, here, right? You search much more thoroughly, and that in turn enhances your chances of finding your segment, assuming that the segment is not, on the other extreme of the nucleus. So there are definitely pros and cons from the biology point of view in this balance of the scaling of MSD with time and the level of ruggedness of your trajectory. Yes?
It seems similar to the optimal foraging mechanism of
I think it is analogous indeed, yes.
I had a comment on the subdiffusion model. So in that case, that portion, let's say, it's saging more truly. It has more chances as well to interact with another parts of for example, let's say a system like chromatin. It will interact with another sections that it may not be the one that it's searching for, and that could end up getting knots, for example, that will make harder for the section to look further for that thing that it's searching for.
Yeah, thank you. So we talked about mean square displacement, a very familiar concept for physicists, but I think we can look at it from new angles in the context of biology, in particular in the context of remote genes finding each other. There is another, I call them diagnostic tool, right? Another diagnostic tool which you can use to learn something about the mechanism of motion, about the environment as well, in which the gene segments move based on the diffusive light trajectories, and this is the velocity autocorrelation function. So velocity autocorrelation, from the name we can tell, well, it has something to do with velocity, right? Correlating the velocities. Autocorrelation means that we want to examine correlations in velocities of the same segment, autocorrelation, the same segments at different times, right? So velocity of the same segment, these velocities are, let's say, time tau apart. Are they correlated? And as we vary tau, that time interval, what happens to the correlations if there are any? Velocity autocorrelation function, as I just said, give us the degree to which the velocities of the random walker, gene segments for example, are correlated when separated by time tau. Now let's recall that in the experiment, velocity is not measured. The experimental output is position as a function of time, xyz as a function of time. But if we have xyz as a function of time, we can, of course, we can calculate the velocity, right? So let's say at time t, this is the configuration of our polymer. This is a particular segment that we are interested in. And at time t, here's the displacement of our segment. Well, we can use the general definition of the average velocity to calculate the displacement by taking the displacement and dividing it by the time interval over which the displacement occurred. I just finished teaching a freshman physics class, so forgive me for these. And so this is definition, right? So let's say our discretization time interval is delta, and we can calculate the velocity by taking the difference in positions and divided by the time interval delta during which this displacement, this change in position occurred. Here's the configuration of our polymer time tau later. Tau, not to be confused with delta, right? Delta is a time interval which we use to calculate the velocity. Tau is a time interval for which we are examining the correlation between the two velocities. So at time t plus tau, tau later, right? Here's the configuration of our polymer, and here's our favorite segment, and here's the displacement of the segment at time t plus tau. And so using the same definition, we take the displacement divided by the time interval delta and get the velocity at time interval t plus tau. And it is these two velocities, the correlation between which we are interested in. And we can then construct the velocity autocorrelation function as the dot product of the two velocities, velocity at time t and velocity at t plus tau. And then we average it. And I will not go, again, through all the details, but just like in the case of MSD, we can examine the time averaged velocity autocorrelation function ensemble averaged or time n ensemble averaged. And so the angular brackets denote the average. Let's sketch some typical graphs for the velocity autocorrelation functions, just some possible scenarios. So this middle curve, so velocity autocorrelation for the time tau, right? So velocity autocorrelation between two velocities separated by time tau. The middle curve, shown in green, is a curve that shows no correlations beyond some very short time. Of course, when tau approaches zero, velocity, well, has to be perfectly correlated with itself, right, when tau is equal to zero. So it has to have some finite value, or we can properly normalize it, so we can imagine it starts at 1. But beyond some very brief time interval, there are no correlations. That's one possibility. Another possibility is positive correlations. And the third possibility is negative correlation. So this diving into the negative value indicates negative correlations. And we can now match these curves to the scaling, anomalous scaling exponent alpha. So normal diffusion, alpha equal to 1, for which mean square displacement grows linearly with time, corresponds to no correlations in velocity autocorrelation functions. Superdiffusion, alpha is greater than 1, is marked by positive correlations, and subdiffusion corresponding to alpha negative, less than 1, not negative, of course, less than 1 corresponds to negative correlations. So we just discussed two very powerful approaches that serve as diagnostic tools, mean square displacement and velocity autocorrelation functions, which we can use. The definitions are very simple, right? So at least in principle. And so we can use these two approaches to try to extract from the trajectories of the genomic segments. Again, all we know is x, y, z as a function of time for each segment, to try to extract the underlying mechanism and hopefully also learn something about the surrounding environment. Are there any questions before we proceed? Yes. Yeah, so the question was about that so far we only had the trajectories of DJ, and this is because DJ, moreover, DJ is on 2 different chromosomes, right? Yes, which will play a role shortly. And this is because at that stage, the experiment was a single color experiment, right? It was only possible to insert a single fluorescent marker of single color in each chromosome, right? And so we just have to imagine another color, let's say red, right, in the same chromosome, marking some V that is far apart along DNA, right? And so we want to know how that V moves relative to the J, right? And in turn, how long it takes them to find each other. We have to imagine that and try to predict that based on what is measured here. And what is measured here is DJ and DJ on 2 chromosomes. So just when you are about to apply this elegant, these elegant techniques, mean square displacement, velocity autocorrelation functions to your diffusive trajectories, you realize that there is a complication. And the complication is evident from the actual video, the live cell imaging. We see that, of course, what we're interested in is the motion of each of the green dots, but the green dots are inside the nucleus inside the cell, and everything is alive, right? It's a live cell imaging. And you can see that the nuclei themselves move quite a bit. They move both translationally and they undergo rotation, right? And that means that the trajectories that are actually recorded, the x, y, z, of course they contain that interesting motion that we want to understand, the motion of genomic segments. But on that motion, superimposed are the motion of the translational motion of the nucleus and also the rotational motion. What do? You are a theorist. You are given the data from cells like this that don't behave very well, and you have your MSD and velocity autocorrelation function. One possibility is to tell biologists, go back and redo the experiment. I did it once and I will never do it again. Do you have suggestions?
You have the center of mass of the entire thing.
Yes.
So that is another information that can be very useful.
So what you can take advantage of is the fact that you actually have 2 dots on different chromosomes, and they move relative to the center of mass, right? So they are inside the same nucleus, right? And therefore, instead of applying MSD and velocity correlations to the motion of individual segments, we can apply them to the motion of the distance between the 2 segments, right? The distance between the 2 DJs on 2 different, on 2 chromosomes. And by analyzing not the change in position of the segment, but the change in the distance, we are automatically eliminating the contribution from the nuclear translation and nuclear rotational motion. Now, of course, one can say, well, then if you stare for quite some time, which is what we did at these nuclei, you'll see there are other types of motion. For example, the nuclei tend to deform, but Well, one can think about ways to deal with that deformation. It seems to me like it's a much more challenging problem than getting rid of rotational and translational motion. But at least in this case, we were able to convince ourselves that the deformation effects are, to first approximation, negligible. But the motion that you definitely cannot ignore, that is just so evident when you look at the video, the rotation and translation, you can eliminate it if you examine the behavior of R, the distance between the two dots.
Can I ask something very quickly?
Yes.
Here.
Yes.
Do you have good information about the depth?
The depth?
Yeah.
Yes. Yes, and this is what I want to emphasize again, because it's not obvious from the video, right, is that the trajectories are three-dimensional.
Yeah.
I think that often the life cell trajectories that are measured are actually 2D, right? So we do have information about Z. Of course, the resolution is much more poor, right, in the Z direction than X and Y, but it really isn't valuable that we have the Z information, because otherwise we would not be able to tell, right, if the two segments appear to be, which is why I think you asked, right, to be closer, if the two segments are actually moving toward each other, or if they appear to be closer just because of the rotation. But yes, we have the information about depth. Thanks for this question. So we can now calculate the mean square displacement not by tracking the change in position of the segments, by tracking the change, the difference in the change in the distance between the two segments, right? But other than that, everything we've discussed so far, including time averaging, ensemble averaging, or both, still holds. And so here's our general scaling for the mean square displacement with time tau, right? Tau to the power of alpha. We can write it now explicitly with the coefficient of proportionality. D is the diffusion coefficient. We're talking about 3D diffusion, and normally you'd expect 6 d tau to the alpha, right? 6 in three-dimensional diffusion, but again, we are looking at the change in the radial component, right, in the distance, and it represents 1 out of 3 degrees of freedom of the motion. So we can actually refer to this as radial MSD, and therefore this 1 out of 3 degrees of freedom results in a coefficient of 2 as opposed to 6. And here's the result. So these are mean square displacements for the coordinate r, the distance between 2 DJ segments. The colored curve, by the way, log-log plot, as we discussed, right, it's the most informative one. The colored curves are the time averaged mean square displacements. So they come, each of them comes from the trajectory from each of the 183 cells. So it's a decent statistical ensemble. And the thick gray curve is the ensemble averaged mean square displacement. In other words, MSD averaged across all these 183 cells. So the first thing we notice is the slope. The slope is not 1. Diffusion is not normal. So we didn't waste our time talking about pros and cons of anomalous diffusion, in particular subdiffusion. For this particular case, alpha is approximately 0.5. So our first immediate conclusion is that the motion of genomic segments is subdiffusive. It's anomalous diffusion. It's diffusion, but it's anomalous diffusion. More specifically, it's a subdiffusion. Next, we have information about time averaged mean square displacement and ensemble averaged mean square displacement. And well, of course, it is natural to expect some inherent heterogeneity among the cells, right? This is not a perfect statistical ensemble. These are live cells. And so perhaps it's not surprising, although I was surprised when I first saw it, but that there is a spread in the vertical offset of these lines. And by the way, let me ask you, so what can we extract from, so the slope gives us alpha. What can we extract from the vertical offset of the graph? Diffusion coefficient, right? We can get the diffusion coefficient just from the vertical offset. Yeah, so there is some spread in diffusion coefficients, but the slope seems to be more or less consistent, and so we can say that, to a very good approximation, the process is ergodic. This is something that we would not be surprised to see in a normal case of normal diffusion, but for such a weird diffusion with scaling exponent 0.5, this is an interesting conclusion.
Can I ask something again? Sorry.
Yes.
Do you have any experimental limitation for the time lag, the maximum time that you take there? Can you wait for longer and see what happens? Yeah.
These actually are short time measurements. So in fact, the trajectories were, I think, at least twice longer indeed. But you know, the later you look, the worse your MSD becomes because you have less material for averaging, in the time averaging, right? But I could have shown you the entire range if
yeah,
you know,
but nothing happens there, right? Or do you have any sort of crossover from subdiffusive behavior to
this is a very interesting question. What you see at very long times, right? Well, I guess we can all guess what would happen if we have no limit on the experimental side of how long we measure. The answer is that MSD should somehow saturate to zero slope because at some point the segments will hit the nucleus, right? Or whatever, maybe there's some confinement even below that. But yeah, so at this point, there is no saturation yet, right? But there was a hint of saturation on the time scale, if I remember correctly, of maybe 400 seconds and beyond, right? But for now, let's focus, this is a very good question, we will turn to it later. For now, let's just focus on this segment of first 200 seconds, which is actually sufficiently long already to give us some meaningful insights about the motion. So comparing the slope, we see that the process is ergodic. Now let's look at what they've, and of course you see faces from time to time, and these are the key people who actually did the work. So Yao Jun was a former graduate student of mine who did all the theory and computation, and she was the one who, when my biology colleagues came to me and asked me if I would be willing to help them work on the diffusion of immunoglobulin heavy chain locus. I said, what? I couldn't get used to this terminology, and so Yao Jun told me, Olga, we have to work on this. This is just too cool. And neither her nor me knew anything about adaptive immune system, so I'm very grateful to her. This is why her face is here. She's now assistant professor in the Department of Physics at Johns Hopkins. Velocity autocorrelation functions. Let's see what we can extract from looking at the correlations, autocorrelations in velocities. Here is a reminder how we do this. So this is a correlation between velocities. Each velocity is calculated by taking the difference in the distance between the segments and dividing it by the time interval delta, right? So, and again, it's important to keep track of these two, not to confuse these two time scales, temporal parameters. Delta is a discretization time interval that allows us to calculate the velocity itself. Tau is the time interval for which we are examining the correlations, right? So for each value of delta, we can calculate the corresponding correlation, and the result is this set of curves. So velocity correlation properly normalized, so everything starts at 1, as a function of the time lag, and the curves of different color correspond to different values of the discretization interval delta. And we immediately notice that regardless of how finely we, how small the time interval we choose to calculate the velocity, for all the curves, there are negative correlations. Here's 0, right? There are negative correlations. All the curves dive into the negative values, which means that there is some kind of anti-persistent behavior. Intuitively, it can be understood as our genomic segment makes a move, makes a step in a certain direction, it bumps into something that pushes it back. And so it means that the most likely, the next step is most likely to be in the opposite direction. And this is what we see in this negative dips. Yes? If you can please repeat it with the microphone.
So
don't you need to have the time lag tau always bigger than the delta? Like it's like you have a resolution of your lag that is smaller than the resolution to calculate the velocity.
Yeah, it's a great question. I can tell you're already thinking about how to actually calculate it. It doesn't have to be bigger, so we can. So we are looking at two velocities, each calculated over delta, right? And they're separated by tau. This is huge tau. This is 0 tau. This is tau less than delta, right? But then related to your question is, what if tau is equal to delta? Because I don't know if you notice from here, but the dip always occurs at tau equal to delta. Can you explain why is this so? This is precisely means that the most negative correlation occurs for 2 consecutive steps, right? This is what it means. So, which is, you move in a certain direction, your next step is most likely to be in the opposite direction. And that is what the curves tell us. Yeah.
Okay, thank you. And I have another question
Yes.
also. So here, you were able to mark these very specific segments, gene segments, the DJ, in the nucleus of the cells. If you could mark different segments there, not two different at the same time, but only one, but it's a different one, and you repeat the experiment, would you expect to see the same patterns in diffusion? Or is there something special about the DJ?
Sorry, there is nothing special about that particular DJ, first of all, right? But I think your question about putting it on different DJs is interesting also in another way, because that would allow us to look into cross correlations, right? But correlations of velocity not for the same segment, but for different segments. But that's just a theoretical physicist fantasy at this point. So the most important conclusion for the velocity correlations is the presence of the negative dips, right? And that is the pushback from the environment. This is how we can interpret it, right? The anti-persistent behavior manifested as negative correlations is a pushback that our segments experience. And one last observation. Let's take this velocity autocorrelation functions and plot them not against the
Olga?
time lag. Yes.
Can we try to understand this pushback as something like you try to see if it fits? Oh no, it doesn't fit, and then it's pushback, or
Fits in terms of like
trying
just to
probing if there is a complex or interaction being formed or not, and once you are within a critical distance, that they would interact or
so we are talking about diffusive motion. So probably those steps are, there's no time for, this pushback as a result of, the segments already being next to each other and deciding whether or not they're going to interact. I think that would require some more time. But I think you are raising an interesting possibility that this kind of, subdiffusive motion is, well, I think it's related to the problem of search, like efficiency of the search that we talked about in the past. Yeah. But here, I think the most natural interpretation is actually something I'll talk maybe a little more detail in a few minutes. We do have a few minutes left. You make a step, and since you are not in a homogeneous aqueous environment, you are in an environment that is filled with springy stuff like chromatin fibers and proteins, right? So you bump into some spring, and naturally the spring pushes you back. So this is probably the simplest interpretation. But what I would like us to do now is to take these velocity autocorrelation functions and plot them not against the time lag tau, but against the rescaled time lag, tau over delta, where tau, again, is a time scale over which we examine the correlations, and delta is the discretization time interval that we use to calculate the velocity itself, which introduces a little bit of, I hate to call that artifact, but it is in a way, right? Because this is we decide how big the delta is, right? So let's divide it out. Let's plot velocity correlations as a function of tau over delta, and this is what happens. All the curves collapse on a single master curve. And this universal collapse is an indication of a property that in physics we know as self-similarity. Self-similarity. Normally diffusive trajectories have this remarkable property of self-similarity. Self-similarity simply means similar patterns on different scales, both on spatial scales and temporal scales. And so this is a property of normal diffusion, right, the fractal nature. But it turned out that our subdiffusive motion, which has this weird scaling exponent and negative correlations, it also has this property of self-similarity. And the collapse, of course, is so good that we can now actually look at it in more details and ask about these outliers and the spread of this collapse. These are also interesting questions. Where does this spread comes from? Is it, from the drawbacks of the experiment or something inside the cells? But the degree to which the curves collapse, I think, is quite stunning, especially realizing that this comes from live cell imaging, the motion inside live cells. So to summarize, our great diagnostic tools, mean square displacement and velocity autocorrelation functions, revealed 4 essential signatures that we will hopefully be able to use to identify the dominant mechanism of motion of DNA segments. And the signatures are, first of all, a subdiffusive scaling exponent. Second, ergodicity of the process. Third, the anti-persistent behavior or pushback from the environment. And fourth, self-similarity or similar patterns on different spatial and temporal scales. And looking at the clock, maybe we have time for, do we have time? Yeah, let's do it quickly, and then we'll pick up tomorrow from here. So how do we extract the mechanism from these trajectories, and especially from the 4 signatures? Things are not as simple, because there is actually a list of mechanisms, of possible mechanisms, that each of which is known to result in subdiffusive mean square displacement. And one possibility is, imagine a Brownian particle that diffuses normally, something that you would, in the end, would get a normally diffusive trajectories. But while diffusing normally, once in a while, similar to what you suggested, it finds some binding partner and pauses there. Binds and pauses, then unbinds, keeps diffusing normally, finds another partner, binds there and pauses. And so this normal diffusion, which is interspersed with pausing and binding events, is known to result into subdiffusive scaling exponents under particular circumstances. The circumstances is that the distribution of pausing times should have a sufficiently long tail, and so then you get subdiffusive scaling exponent. And we can imagine this happening in the cell, right? The normal diffusion, which is interspersed with pausing and binding events. Another possibility is diffusion that would otherwise be normal, but diffusion occurs through obstacles, like hard wall-like obstacles. And when the concentration of obstacles is sufficiently high, then the resulting motion has subdiffusive subdiffusive scaling exponent alpha. And another possibility is diffusion through obstacles, but the obstacles are not, hard wall-like obstacles, but they are springy. And this is a diffusion in viscoelastic environment, which is also known to result in subdiffusive scaling exponents. Yes?
Sorry, so the fact that there is an attachment never plays a role in this?
That what never plays?
The fact that you have some connection between the particles is not considered like a constraint also?
Thank you for raising. This is actually the crucial aspect. The fact that we're talking about polymer, right? Exactly, yes. In fact, it's so obvious that it's easy to forget about it, right? We shouldn't forget. Thank you for reminding. This is, of course, an excellent point. This is not the whole list of possibilities. There is another possibility which I really wish I didn't have to think about, but I have to, and this is what is known as localization error, basically the finite resolution with which the position of a segment can be measured, right? And so it has been shown by some careful researchers who like to look into the things that localization error, unfortunately, can mimic subdiffusive scaling exponent, even though the diffusion otherwise can be normal. And so the signatures of motion that we've discussed so far are consistent, fortunately, only with one of these mechanisms. And let's talk about it tomorrow. And the fact that we're talking about not isolated particles, but the fact that they're actually polymers in a monomers in a polymer chain is going to play a crucial role in the resulting dynamics. I think we already passed the time, so I just don't want to push it any further. We'll pick it up tomorrow. Should we have any questions, or should we just go to lunch, whatever the organizers think?
I think we have time for a few questions.
Thank you very much for the lecture. About that slide we presented the mean square displacement, we talked about the definition of velocity. I don't know, maybe you can show that slide? I have a doubt about this definition when we're talking about...
You have a doubt?
Yeah.
We definitely should go back
Please.
if you have doubt.
Yeah,
Yes.
precisely.
I like doubts.
When we're talking about, for example, subdiffusive process in short times, can we define very well velocity in this case? Because when we are thinking about the mean square displacement in short times in this process, if you take, for example, the square root for the mean square displacement, it divided by time, the function will diverge.
Yeah.
So the velocity diverges.
Definitely, yes, but in experiment, we can take time to equal 0, right? So tau has some smallest value that we can examine, which is why we are operating with average velocity when we calculate velocity and then velocity autocorrelation, not instantaneous velocity, right? So in practice, we never have that problem. In theory, of course, we do have to worry about that thing, so it would be a good point, yeah.
I do have a question. It's very striking, this self-similarity. And did you actually fit the exponent for the scaling collapse? Or did you just try something that made sense, tau over delta?
So first of all, thanks for asking. There's 0 fit here, right? There's no fit, assuming any parameters. This is parameter-free plot where we just took all these curves and replotted them. The curves are from the trajectory, so nothing is fitted, and replotted them against tau over delta, right? So this is what the data tells us. Now, I think then the question is, either a curve we can either fit or, well, either a curve preferably derived consistently, right, from some theoretical considerations that we can fit to this behavior. Yes, there is a curve, yeah. It's a separate interesting story, but basically the question, for example, of what kind of dependence this is, and also how fast it decays to 0, it's something that actually is controlled by alpha. It's controlled by the scaling exponent alpha, but from that fit we can also learn a great deal about this. Yes, it's a great question.
Thank you for the talk. I was wondering, have you considered the mechanism of diffusion with the stochastic resetting as a mechanism to find this V target? Meaning that every amount of time the particle searching for the target resets to the original position?
On what time scale do you want it to reset it to the original position? So what I plan to talk about tomorrow is how we simulate this motion. And so maybe it's also a useful way to sort of imagine what the particle, not the particle as we were reminded, the monomer of the polymer of the chain is doing. So it starts at some initial position, right? Then it is moving according to some equation, which we are going to establish tomorrow, right? Based on the mechanism that we will have identified. And then we just let it do whatever it wants to do according to that equation and ask how long it takes that particle to find another particle. Or if we are thinking in terms of the relative distance between the two segments, how long it takes the distance to become 0. or since we have very careful people here, we can't quite demand 0, right, because of the experimental resolution. So we have to actually think, which is non-trivial, about the threshold value of R, below which we can say our segments have found each other, right? And then, once they have found each other, then we do the reset that, like you suggested, we put the particle back and do it, let it, do another realization of the random walk, right? And measure the time again. And so from many, many, hopefully such measurements, we can find the mean time, also known as the mean first passage time. Yes. And then there's a related question about when you reset, actually when you start your simulation, what can you or should you assume about the initial position of the particles, right? Or in your statistical ensemble, how do you want to distribute your particles? So there are many subtleties.
Question.
Yes.
When you talk about the universal collapse of this, can you relate to somehow a kind of surface tension, or eventually you have charges associated, but you know that it's going to be, it's not a charged system, though it's inhomogeneous. So could you
I think what you're saying is something that would make me not expect a universal collapse, because there are so many complications, right? So actually the fact that the system does exhibit self-similarity is striking to me, right? So it basically means that if you rescale your time lag by the discretization interval, then the difference between different curves disappears, right? So there's this universal behavior, which is something you would get for normal diffusion, right? And it's a remarkable property of fractals, is that, this stunning patterns, that they actually have similar patterns on different scales. But we are here talking about very strange subdiffusive motion, and so the fact that it does exhibit self-similarity is a surprise, and it honestly is a surprise. So it's not, I would say, it's not something that we would have to attribute it to. It's something that is happening despite all the complexities of motion and the environment inside the nucleus.
Perfect. Well, last question.
First of all, thank you so much for the lecture, and I have a question regarding the experiment. So for the points, I guess that they are of a certain size, so in kilobases. So how much will it change the understanding of the motion, the diffusion, if we take a finer resolution for each of the fragments of DJ and B?
Is the question about the temporal or spatial resolution, if we have no...
Yeah, if we use like a finer spatial resolution for each of the bits, or
finer resolution, is it a question?
Will it change our understanding on the diffusion of the motion, or
of course, our hope is that our conclusions would not change, right? Because otherwise, the finite resolution would be a source of artifacts, and which is related to a point that I mentioned very briefly because of the time constraints, but maybe I'll try to remember to return to it tomorrow, is that the fact that the resolution is not as good as we would want it to be, sometimes introduces artifacts that can basically fool you. In particular, they can mimic negative correlations, even though the real motion actually does not have negative correlations, right? And so this is something we have to think about carefully, and specifically about this point, the way that localization error, that finite resolution introduces the fake subdiffusive motion, is it only manifests itself at very short times, and actually the fact that the depths of the deep, right, it is nearly constant, to a good approximation, nearly constant here, but it has this particular behavior at short times. It goes a little bit up, right? It is actually a signature of localization error, which tells us that we do have that artifact and we are aware of it, but it only manifests itself here. And in particular, if the whole observation of negative correlations was just due to that experimental artifact, we would definitely not get a universal collapse. So this is something that we can extract from the self-similarity, is this is not just due to finite resolution.
Thank you so much.
We reconvene. So let's thank our speaker again.
Thank you.
And we reconvene at 2 o'clock, okay?
Okay, so now basically we have lunch break, right?